What I would really like to share with you this afternoon are some of the ideas that we have on the origins of life, but most especially some of the unknowns that we have in a field that we now realize that is absolutely valid scientifically, but also has many implications from the philosophical viewpoint in terms, for instance, the fact that we do not have a very good definition of what life is. Hence, this immediately poses a problem if we want to try to understand either in the fossil record or under laboratory conditions what are the traits that we can actually identify in our chemical models as corresponding to an actual cell or an actual living being. So the first thing that has to be said is that we know that life is a very ancient phenomenon on the planet. And this is a very important realization because it immediately links the phenomena that preceded the appearance of life with the actual astronomical and planetological conditions of the planet. We know that the Earth was formed together with the rest of the solar system 4.610 to the 9 years ago. And we know that at the very beginning, the Earth was not only very hot, but was also undergoing a major number of collisions. The rest of the solar system nebula that was collapsing and was actually provoking a very intense collision between small bodies and the primitive Earth. So clearly during this time, we have the feeling, which may be wrong, but we have the feeling that the planet was not hot, was not cold enough as to be able to sustain cold water on its surface. And it's very difficult for us, biologists, chemists, to imagine how life could appear in the absence of liquid water. All our models suggest that this collisional intense phase ended around 3.810 to the 9 years ago. And if we go backwards from the present to the past, we have different kinds of fossil records, some of which are disputable, that suggest that life indeed will appear on the Earth as soon as the Earth cooled down. Some of the nature of these records are contested. For instance, my friend and colleague Bill Shaw from the University of California at Los Angeles has investigated a number of small structures that appear extremely rich in organic compounds, but some people claim that those structures are actually not the remains of living cells, but artifacts made due to crystallization processes or condensation of inorganic material or whatever. On the other hand, we can actually examine the isotopic record of this organic material, and we see what we call isotopic fractionation. In other words, we do see that the tendency of living systems is to go always for the smallest form of carbon, for the less heavy form of nitrogen, of oxygen, of sulfur, and so on, in contrast to what happens with most inorganic processes. And therefore, the fact that we see this isotopic fractionation in rocks or in remains that are about 3.4, 3.5, 10 to the 9 years ago, suggests that we are actually seeing evidence of biological activity. In general, most of us working in the field accept that by 3.4, 3.5, 10 to the 9 years ago, there was already a highly diverse microbiota inhabiting many different parts of the planet and exhibiting a wide variety of different kinds of metabolism. Not oxygenic photosynthesis yet, that came later, but clearly we see fixation of sulfur, we see production of methane and so on indicated by the isotopic record. The real problem is indicated here by this question mark, because we have a gap in the geological record in which we have absolutely no sediments that can tell us what was the pH of the primitive oceans, what was the composition of the atmosphere, what was the temperature of the ancient hydrosphere. So under such conditions, it's very difficult for us to actually propose a reasonable model of the environment in which life appeared. Clearly, life must have appeared prior to 3.5, 10 to the 9 years ago, but later, most very likely later than 3.8 to the 9 years ago. Life appeared very quickly and under conditions that we certainly do not know. Now, one of the very first to propose How this could be was Alexander Ivanovich Oparin, a Russian, Soviet, if you wish, biochemist in the 1924. But every theory that we have, or every hypothesis that we can propose on the origins of life, has to face a number of problems. In other words, our understanding of the origins of life is limited very strongly by the fact that we lack geological evidence of the prebiotic environment. We don't have an idea of what was the nature of the planet at the time. We don't have a fossil record of prebiological systems. The oldest fossils that we see are actually very complicated, so that clearly implies that that must have been preceded by simpler systems of which nature we know nothing about. And finally, we lack an all-embracing, generally agreed upon definition of life. And this is not a minor problem, because for biologists trained a certain number of years ago, if you have synthesis of proteins, you already have something that's alive. For more recent life scientists, if you have something that is catalytic, like RNA, it's already alive, even if you don't have protein biosynthesis by then. So clearly here we have a problem that complicates our perception of what the first life form may have been. Having this in mind, one can actually look at a different perspective of the proposal made by Oparin in 1924, and that was refined in a book that I believe is one of the masterpieces of evolutionary biology published in the 20th century. It was published in Russia in the Soviet Union in 1936, and was translated into English in 1938. And here you see actually the cover of the book, in which Oparin actually suggested, after a very detailed analysis, being a biochemist, a very detailed analysis using information from the astronomers, the geologists, the philosophers, the physicists, the biochemists, and so on, that the early Earth was lacking free oxygen and therefore had what we call a reducing atmosphere, what astronomers call a reducing atmosphere. And Oparin really realized very quickly from all the experiments done in the 19th century that if you have an atmosphere that is lacking oxygen, you can actually synthesize without any problem organic compounds. He proposed that these compounds were washed into the primitive oceans and formed what we call the primitive soup. And at the time, les derniers cris de la mode, to understand the nature of cells, were quacerbates, a physical chemical system that is a gel-like structure. So Oparin rapidly proposed that the first ancestors of extant cells were actually quacerbates formed by the aggregation of the polymers that had been accumulated in the primitive soup, and the biochemical reaction between the molecules forming the quacerbates actually led to the first bacteria, which are the oldest living beings, and they were primordial in the sense that they were the first entities to be alive. They were anaerobic because they had appeared in an environment that was free of oxygen, and they were heterotrophic because they were nourishing themselves from the organic compounds that had been, from which they had formed. So this is basically the idea behind the so-called heterotrophic hypothesis. Now, if you look at this scheme, you rapidly realize that nucleic acids are absent, which is very easy to understand. In 1936, if you go to Google, you will see that James Watson was in 5th grade of grammar school. So there was absolutely no way of imagining the role of the double DNA helix at the time. And furthermore, as late as 1942, some very distinguished evolutionary biologists like Julian Huxley, one of the proponents of the neo-Darwinian synthesis, was actually arguing that bacteria do not need genetic material. So the fact that Oparin is not mentioning DNA or RNA in this context is simply an indication of the prevailing ideas in a very large group of biologists of the time. The proposal by Oparin was produced in very complicated sociological and ideological conditions. Oparin at the time was living in the Soviet Union. In 1936, it was just prior to the Second World War. The Soviet Union was undergoing what we can call in many ways a cultural revolution that is very easy for me as a Mexican to understand, because, for instance, if you look at the art of Frida Kahlo and Diego Rivera in Mexico, these were produced immediately after the revolution. So there was like an explosion of creativity in many areas of science, of culture, of art, and so on. And in the case of Oparin, what we see is a social environment that was clearly promoting a very, very laical, a very secular explanation, evolutionary explanation for the nature of life. One thing that is also worth mentioning is that my very good friend Joe Felsenstein, one of the most distinguished evolutionary biologists in the United States, when he saw this picture, immediately told me that this was made by a very famous communist artist in the United States who was actually making drawings for books for the general public. So you see right away the connection between a certain ideological position and the approach to the origins of life that was proposed by Oparin. That led, and we can discuss it afterwards, to a number of major problems even today. For instance, there is a general misunderstanding, and I have to right away say it's wrong, that Oparin proposed his theory because he was pushed to do so by the Soviet Communist Party. That's absolutely untrue. He had very sincere intellectual scientific interest, but the general atmosphere was certainly such that you could indeed propose this kind of evolutionary ideas. Now, clearly, the theory, we have a number of experiments and observations that allow us to go beyond these early proposals. And I think it's fair to say that nowadays, if we compare what Oparin proposed in 1936, 1938, to what we see today about the origins of life, I think it's valid to say that we still accept the idea that life was preceded on the planet by the synthesis and accumulation of organic compounds. I will review how that could have happened. We recognize that RNA molecules actually play a key role in a number of processes, both in terms of regulation, storage of information, catalytic activity, and this has led us to propose to accept the idea of an RNA world, and the activities that we see in RNA world, including the fact that the ribosome in the catalytic site of the ribosome has no proteins, allow us to propose that protein biosynthesis started in an RNA world. And therefore, we can quite easily understand in terms of biochemical evolution, natural selection, acting over living systems, that eventually we get to cells that have proteins, that are endowed with proteins, RNA, and DNA. So what you see here is actually the modern adaptation of Oparin's ideas, which mean that epistemologically, the theory was what we call open. It could accept different kinds of evidence without coming down, without the whole structure coming down. A little bit like Darwin's idea of natural selection. You know that Darwin didn't have a good theory of heredity, but nevertheless, it was so well sustained, the idea of natural selection, that it could incorporate the modern discoveries of genetics without any major problem. The other thing that I want to emphasize is that when we look at Oparin's early proposal, what we are actually seeing here is the discussion of a program of interdisciplinary and multidisciplinary research. Oparin is saying that we cannot explain the origins of life by assuming the sudden appearance of a living entity, but that you require information from the astronomers, from the planetologists, from the chemists to understand the synthesis and accumulation of organic compounds. We now realize that we need to understand the molecular biology of RNA to understand what we mean by the RNA world. And of course, this transition requires recovering an ancient tradition that was lost around the 1960s on evolutionary biochemistry. But this will be basically the guiding line of what most of us agree led to the appearance of life on Earth. Now, if you look at the theory, both the original proposal and the current one, you see that what Oparin or many of his followers are saying is that if you want to have life on Earth, you need first the synthesis and accumulation of organic compounds on the planet. And now we are quite confident that the synthesis of organic compounds of biochemical significance actually took place prior not only to the origins of life, but actually long before the origin of the planetary system itself, long before the origin of the Earth. And the evidence comes from astronomers. Astronomers now have an extraordinary sophisticated set of observations that allow us to see how the interstellar cloud we can see from simple molecules that are probably formed in the outside part of red giants and stars that are on the late stages of evolution in the main sequence. These molecules are actually expelled to the interstellar medium. We know that in the interstellar medium, where we have the clouds of gas and dust, there is a very complex chemistry going on the surface of what we call interstellar grains, and sometimes these clouds actually can collapse and give origin to planetary systems. This was an idea that was proposed a long time ago. Actually, the first to speak about the idea of solar nebula collapsing and condensing and giving origin to a solar system was Immanuel Kant, the philosopher, and then 10 years or so afterwards, Le Marquis de Laplace actually developed a quantitative model of this idea. And you French people know much better than I do, I'm sure, the famous anecdote that when the Marquis de Laplace showed his book on celestial mechanics to Napoleon I, Napoleon I, who could be very mean, look at the book and say, Monsieur le Marquis, I do not see the word God in your book on celestial mechanics. And the answer of Laplace, I think, is perfectly revealing of the attitude of the laïcité that the French and the Mexicans say. The answer was, Monsieur l'Empereur, it's a hypothesis that I do not need to appeal in order to explain my model. We don't need, in fact, nothing but pure laws of physics, chemistry, to understand the evolution of the solar systems. And now we know that we have a huge number of planetary system, a huge number of exoplanets that actually validate this scheme of the origins of life. The condensation of the bodies in the disk turning around these central bodies, in the case of the solar system, around the sun, eventually led to larger and larger bodies that accumulate due to gravitational effects, and the final consequence is, of course, the planets that we see today in our own case or in exosystems. What evidence do we have that indeed the early Earth was being subject to the bombardment of many of these bodies? We have the moon. Of course, if we look at the Earth, we know that the Earth is quite active geologically. We have lost the record of the early events that took place when the Earth was young. But if we look at the moon, we are actually looking at a surface that became frozen, if you wish, about 4 billion years ago. 4 billion years, I'm always confused by the billions in French, in the billions in the United States and the billions in Mexico. We tend to have an inflated view of years in Mexico. So if you look at the moon, you see a huge number of craters. Most of these craters are actually due to collision with smaller bodies. And we know that the moon and the Earth are so close that if there were collisions, there were bodies colliding against the moon, clearly there must have been the same amount of bodies or even greater condensing or colliding against the primitive Earth. So one thing that has to be said is one of the major achievements of planetary exploration is the realization that collisions were an important part of the early history of all the bodies in the solar system. In some cases, like the Earth, that history has been erased by geological processes. In some cases, like the moon, we still see that record, and we can extrapolate from there to our own planet. What kind of bodies actually collide against the Earth nowadays, and what were the nature of the bodies that were colliding against the primitive Earth? Well, we can actually, in some cases, reconstruct the orbit, for instance, of the Low City, the Peekskill, the Innisfree, and the Pribram meteorites. And if you look at the points in which they collided with the Earth, when the Earth was in these different parts of its orbit, you can see that we have enough data to actually reconstruct the orbit followed by the meteorite before colliding with the Earth. And we always see in one of the focus of this ellipse, we are in the zone of the asteroids. And we know the asteroids extremely well. We actually are landing now on asteroids without any major problem. And if you look at the asteroids, which are these small bodies, planetesimal remains of what actually formed from the planets, you see that some of them are highly eroded, and clearly just by looking at this photograph, you can realize that you have different kinds of material with different levels of resistance to erosion in the interplanetary space. In some cases, we can see, for instance, one of the moons of Mars that is clearly an asteroid that was captured. Phobos and Deimos were captured by Mars during its early the early history of the solar system. So what we really can picture is the asteroids circling around the sun. Eventually they can collide, and the pieces are sent in all directions, some of which actually come towards the inner part of the solar system, and the Earth can capture them. And in some cases, we are lucky enough to actually see the pictures of the meteorites, of the remains of these small bodies coming into the atmosphere. Now, it's obvious to all of us, but let me underscore a point that is important. We see this light just because we see the body being heated up, emitting light by the friction when it collides against the atmosphere of the Earth. It's a point that I have to return right away. We were very lucky in that in 1969, a meteorite fell in Australia, in a region called Murchison. It was September 1969, and we actually know the hour. It collided at 10:55 in the morning. And we know that it took place on 10:55 because the pubs opened in Australia at 11. So there was a huge crowd of Australians, which can be very heavy drinkers. There was a huge crowd waiting for the pubs to open. They realized that the meteorite had fallen. Immediately, the people went to collect the remains of the meteorite. Actually, you can see here the town of Murchison, and you can see the places around the town where the people found the meteorite. They found close to 100 kilograms of meteorite, and by reasons that are very clear to me as a Mexican, not so clear to some of my colleagues, of those 100 kilograms of meteorites, about 98 ended up in the United States. But the meteorite was there. It was collected immediately after it fell down. It was therefore basically free of any possible contaminants. And the interesting point is that the meteorite, when it was analyzed, it could be studied under laboratory conditions that have been designed for the study of the moon samples. In other words, they can be studied under very clean conditions that avoid contamination by biological material. The first surprise by the meteorite is that it turned out to be 4.6 10 to the 9 years ago. It's exactly the same age as the solar system. So we are witnessing, if you wish, a frozen sample of the material from which the planets form. The other point that I want to emphasize is if you look at the meteorite, and it's perfectly clear in this part, you can see that the outer part seems like a burned baguette, like a baguette that remained for too long in the oven, was burned down, but you can see that the inside, like a baguette, is really not affected. And that's a very important point because if you burn a baguette, you can actually eat the inside without any problem. If you burn this type of meteorite, because they are made of a stone, the stones are very bad conductors of heat, so therefore it's only the outer surface of the body that is affected by the heat when it traverses the atmosphere, but the inner part of the meteorite is essentially unchanged. So what we do is we take samples of the inside of the meteorite and we analyze them chemically. It sounds very impressive, but I'm sure that many of you in this room have gone to see your levels of sugar or cholesterol in a medical laboratory, and exactly the same procedures that are used to analyze blood samples are exactly the same procedures that we use to analyze meteorites, only it sounds much more sophisticated and elegant and good to impress friends if we want to. When we analyze meteorites, we discover that there is a huge fraction of polymer whose actual structure is not known. It's very complicated. We know it has nitrogen, oxygen, carbon, sulfur, hydrogen, but we do not know the actual chemical structure of the molecule. It may be a set of different kinds of molecules, but then we find a small fraction in this Camembert diagram. We find a small fraction of organic compounds that are soluble, and we see that many of them are carboxylic acids, but we also see dicarboxylic acids, the same ones that you see in the Krebs cycle are found in the meteorite. We see about 90 different kinds of amino acids, amides, aliphatic hydrocarbons, aromatic hydrocarbons, aldehydes, alcohols, purines, amines, and a few other compounds. So the meteorites are telling us that we have an extraordinary diversity of organic compounds clearly coming from outer space, because, for instance, we see the racemic mixtures of amino acids, the L and D forms in the same proportion, and clearly they are telling us also that they are not the outcome of biological activity. Given the age of the meteorites, in many ways we can consider a meteorite as a sort of chemical fossil of the events that took place in the early solar system. And what this is really telling us is that if we see this kind of synthesis going on the solar nebula, it's very reasonable to assume that equivalent processes took place on the primitive Earth. Now, together with some colleagues, I have been engaged in the analysis of meteorites, especially amino acids. And here you see, for instance, in red, the different kind of aliphatic amino acids that we reported, confirming other reports in a sample of the Murchison meteorite. Then we actually did an experiment repeating a Stanley Miller original experiment with a simple atmosphere with methane, water, ammonia, and hydrogen. And you actually see that the correlation is not so good. But then we decided to add sulfur to the sample, to the mixture, and you can see how we have a superb correlation between the amino acids that we're synthesizing in our simulation of the primitive Earth with the amino acids that we see in meteorites for this particular case. So meteorites are validating our models and vice versa. We have a very reasonable chemical explanation for the presence of these compounds on the meteorites. Now, unfortunately, we don't have a sample of the primitive atmosphere. We do not know what was the actual nature of the primitive atmosphere of the Earth. But in 1952, Harold Urey, who was a very distinguished chemist, an exceptional person, he was actually one of those scientists the United States has every reason to feel proud of, because among other things, he refused to denounce his colleagues against the McCarthy commissions and so on. Urey had been engaged in research at the Manhattan Project, decided that he was absolutely horrified with the consequences of nuclear weapons, and decided to move as far away as possible from the type of investigation in chemistry. So he decided actually to go to cosmochemistry, and he was very concerned about the chemistry of the early solar system. And in 1952, he published a paper in the Proceedings of the National Academy, which is a very elegant paper for the simplicity of the assumptions. Urey say, okay, if I want to understand the chemistry of the early solar system, of the atmosphere of the primitive Earth, what I have to realize is that the Earth was formed from that nebula that Kant, Laplace, and many others have proposed. So I will look into the composition, the elementary composition of that nebula, say Urey, and what I see as the most abundant element is hydrogen. So clearly, he said, hydrogen must be the determining component of the chemistry of the other elements. The second most abundant element is helium, but helium is a noble gas. And you French know what to do with nobles. You tend to chop their heads off, and because they don't mix with the rest of the people. Helium will not mix with the other elements, so he did not have to worry about them. And then he said, well, if I have a carbon with hydrogen will give me methane, nitrogen with hydrogen will give me ammonia, oxygen with hydrogen will give me water, and the rate constant in this case is 10 to the 41, which is huge, a very favorable reaction. sulfur with hydrogen will give me hydrogen sulfide, and I will have very little CO2. Urey was part of the chemistry department at the University of Chicago. And in 1952, he presented his results in a seminar to the students, the graduate students, the professors, the researchers. And at the audience were two students, both of which eventually told me exactly the same story. One of them was Carl Sagan, the astronomer. And Carl Sagan heard Urey present this idea and was fascinated. And in the back of the room was Stanley Miller, who had just joined the chemistry department to work for his PhD in chemistry. And again, the same story was that when Urey finished his seminar, somebody got up in the room and asked him, Professor Urey, are you aware that there is a Russian chemist that is proposing the same kind of atmosphere to explain the origins of life? And Urey said, no, I know nothing about it, but it will be very interesting to do research in this direction. Eventually, Stanley Miller went to Urey and asked him to be his advisor so he could work on research in this type of atmosphere to understand the origins of life. And Stanley Miller was accepted eventually by Harold Urey as a student, and he very quickly designed a very simple apparatus that I'm sure many of you have seen in the books. It were two flasks connected by this tube. Here you had boiling water, and the vapor came up and mixed with hydrogen, ammonia, and methane in the upper flask. We had electric discharges to promote the chemical reaction, and when the products cooled down, they were collected down here, and they were analyzed without any problem. As you can see in this photograph of Stanley Miller after he had published the experiment, the apparatus was indeed quite small. It was a very inexpensive experiment, but very elegantly designed. And what I found fascinating is how the standards have changed, because the upper flask was covered with asbestos, which no one in amiant, which no one in his mind will use today under laboratory conditions. But most importantly, you can see that at the time, the students knew how to dress when they entered the lab. They had white shirts and ties and so on. And somehow the dressing standards for students have changed. In any case, Stanley performed the experiment. He was absolutely happy with the results. I several times asked Stanley, what did you do with the original apparatus? And the answer was consistently the same. I threw it away because it was very difficult to clean. So this classical experiment ended up in the La Poubelle of the Department of Chemistry of the University of Chicago. But Stanley Miller was so precise in his description of the experiment that we can repeat it without any problem. And here you see a repetition that was done in the lab of Bill Schopp at the University of California, Los Angeles. Here you see the experiment at time 0. You can actually see the spark fly between the two electrodes, and only 5 hours afterwards you can see how the internal composition of the gases of the environment has changed. Well, what Stanley did was to take a sample, did paper chromatography, and very soon was able to discover the presence of glycine, aspartic acid, alpha-alanine, beta-alanine, alpha-aminobutyric acid, and a few other compounds. He was absolutely delighted with the results, showed them to Urey, and Urey said, let us publish the paper right away. And let me tell you a small anecdote that I think is highly revealing of the moral standards of Harold Urey. Harold Urey told Stanley, you should write the paper and we can send it to Science. So Stanley wrote the paper, put his name first, and then added the name of Harold Urey in the manuscript. He then gave the manuscript to Harold Urey, and the first thing that Urey did was to erase his own name. He said, it was you who had the idea to do the experiment, it was you who did all the work, it's you who deserve to be famous, not me. So it really shows you the concern that the people had in doing science, not for the sake of fame, but actually just because they had the intellectual curiosity, which I think is a lesson that we should all keep in mind. Well, the experiment was published, and Stanley became instantly famous, and it was actually for him very complicated because after only a few months in the graduate program, he already had the paper for his PhD thesis finished, which put him in a very difficult condition. several years ago, I was doing a sabbatical at the laboratory of Stanley Miller at La Jolla in California, and one morning he came in very excited, and he had a fax in his hands. And the fax that he wanted to show me was an offer from the Museum of Natural History in South Kensington in London, and they were offering him a huge amount of money for samples of the original experiment. And when Stanley showed it to me, I said, but clearly you don't have any samples of the original experiment. And he said, yes, I do. And I said, I wanted to see them. So we went to his office. He climbed over huge amounts of books, photocopies, theses, and so on, produced a box. From inside the box, there were little boxes, and from one of these little boxes came a small vial in which actually Stanley had kept, since 1953, some of the original material he had synthesized. I was very moved, and I asked Stanley, can I make a photograph? Sure, here you have the photograph. Can I actually see it? Can I hold it in my hands? And Stanley gave me the vial to hold it. And then I asked Stanley, are you going to sell this to the museum in London? And he said, no, by no means. And I told him, but Stanley, this has to go into very good hands. He said, yeah, it will go into good hands, and he gave it to me at that point. So I was deeply moved. I had now with me the samples of the original experiment done for Stanley Miller, so I just put them in my pocket, and when I went to Mexico, I took them with me, and when my students behave properly, I show it to them, and they are quite happy, or if they have an examination, they take a look, and they present themselves, whatever. So perfectly happy. A few years later, Jeff Beda, who inherited the lab of Stanley Miller, and myself were invited to give lectures on the origins of life in the United States, in Texas. When it's occupied Mexican territory, it's not really the United States. But we were invited to give a seminar, and the night before the seminar, we were showing to each other the slides, the figures we would be explaining, and Jeff Beda asked me, what do you have here? And I told him the nature of this material, and then Jeff asked me, why don't we reanalyze the samples using the modern techniques? And I thought it was a very good idea, but immediately we were faced with a problem. You can picture a Mexican going across the border with an unknown chemical compound in white in his shirt. It can lead you to some problems. So for about a week or so, we were wondering what should we do. But then I recall a British spy movie. So I bought a book, I made a hole in the book, I put the vial inside the book, and then I sent it to him by DHL or Federal Express. And when Jeff Beda had the samples in his hand, then I flew and we did the first analysis. And the first analysis were very, very encouraging, because you can see here, if we have the sample non-hydrolyzed or hydrolyzed in acidic conditions, you actually see glycine, which is very abundant, but you right away can see beta-alanine, which is not so abundant in biological material. So there was an indication that that the sample had not been that heavily contaminated. And there we were perfectly happy to see the peaks there are telling us that we have racemic mixtures of serine, racemic mixtures of alanine, so they clearly indicated that the sample was worth analyzing and it was not heavily contaminated. We reanalyzed the samples, and here you have in the upper chromatogram and in the lower chromatogram the addition of all the compounds that we discovered. We actually discovered over 40 different compounds, where Stanley Miller had reported only 5. This doesn't mean that Stanley was wrong. It simply means that we have much better analytical tools to actually understand the chemistry of the primitive Earth. And that we thought was extremely encouraging and certainly validated the Miller experiment. But one point that has to be kept in mind is that 1953 was an excellent year. It was actually a very good year for wine in France as well, but it was an excellent year for life sciences, because it's the year in which Watson and Crick proposed the double helix model of DNA, and it's the year in which Stanley Miller demonstrated the synthesis of organic compounds. I want to link the two proposals, the two papers, because this really means that biology ever since enter a highly reductionist, valid but reductionist road in which everything requires to be validated in the eyes of many by molecular explanations. You cannot attempt to explain the origins of life if you do not appeal to the results coming from molecular biology. And this actually led to a very important issue because all of a sudden the people started criticizing the models of Oparin that was proposing a theory prior to the development of molecular biology. But now we have a new theoretical framework. Epistemologically, historically, the situation was changed completely. If you want to have a reasonable explanation of the origins of life, at some point you have to address the issue of the nature, the chemical nature of the hereditary material. And quite surprisingly, a major result came from the laboratory of Juan Oro, a Catalonian chemist that left Spain because he was absolutely oppressed by the intellectual, so to say, atmosphere of Franco dictatorship, went to the United States and started working on the origins of life. And then he realized that in a very simple way, you could actually condense hydrogen cyanide, and hydrogen cyanide actually gives you the dimer, then the trimer, then the tetramer. You can add formamidine, and then you can form the imidazole group, and with more formamidine, you can form adenine. And a few years later, Jim Ferris and Leslie Orgel demonstrated that if you have the aminomalonitrile, which is the tetramer of hydrogen cyanide, it actually absorbs light both in UV and in the visible region and undergoes a photoisomerization to give you the imidazole group, and the addition gives you adenine. So you have this extraordinary experiment that was actually showing in the 1960s that you could form one of the components of DNA under primitive condition. And all of a sudden, all our ideas on the origins of life appear to be validated by this type of experiment. Now, this experiment is still seen by everyone working in origins of life as an extraordinary, elegant example of how you can get a very complicated compound that participates in bioenergetic processes, in hereditary processes, just by the simple pentamerization of hydrogen cyanide. And this was so important that when John Oro retired and went to back to Barcelona, the Spanish were absolutely happy, and they produced an stamp for the mail that was actually honoring the reaction that five hydrogen cyanide molecules will polymerize and give you adenine. And on the 24th of June, which was the birthday of Joan Oro, one day he got a call in Barcelona. He couldn't answer the phone. He was ill already by the time. But his wife answered the phone, and it turned out that they were calling on behalf of the King of Spain. And at first they thought it was a joke, but then it turned out that the King of Spain decided to make John Oro a Marquis because they were so proud of his achievements. So Joan Oro became Joan de Oro and was now a Marquis. Now, as you know, if you have a title of nobility, you have the right to have an heraldic stamp. You can have your seal. And John Oro was the first Marquis de Oro, so he had the right to design his own stamp, and he did so. And here you have what John Oro decided. The crown represents the fact that he's a Marquis. This band represents him with his 4 children. In an act of modesty, beyond belief, he represented himself as the son. This is a small hill of which the Catalonians feel very proud, called Montsec, the dry hill. I never understood why they feel so proud about it. But in this part of the seal, you can see that actually is the structural formula of adenine. It's the only nobility stem that has a structural chemical formula. And it's fascinating because you can see 1, 2, 3, 4, 5 hydrogens, 1, 2, 3, 4, 5 carbons, 1, 2, 3, 4, 5 nitrogens. 5 hydrogens, 5 carbons, 5 nitrogens. Adenine is nothing but a pentamer of hydrogen cyanide. And if you work on the field, you can even become a noble, which is something some people may find attractive. I couldn't care less. But the basic thing behind adenine is that we know that adenine actually binds by 2 hydrogen bonds to uracil, which is a compound that we can also make quite easily under prebiotic conditions. And it's a compound that we actually see in meteorites as being quite stable. Because of the chemistry of pyrimidines, very likely, if we are looking at uracil, that means that you can also have cytosine, but the cytosine deaminates very rapidly and goes into uracil. But the fact that we can form these compounds quite easily, and the fact that we can polymerize them, although we do not understand the nature of the backbone of the first genetic material, is telling us that indeed not only the components of proteins, but also the components of nucleic acids could be present in the primitive Earth. The basic point that I would like to emphasize is that once we have the possibility of hydrogen bonding, you automatically have the possibility of molecular replication or the formation of structures, highly stable structures, as in the case of DNA, or very complicated three-dimensional structures, as in the case of RNA. And as we will see now on, this can be actually extremely important. I will not go into all the details of prebiotic chemistry, but suffice to say that we have experiments done in the lab. We can analyze meteorites, and all the evidence that we have strongly suggests that before the origins of life, there was a wide array of organic compounds of biochemical significance in the primitive environment. There were many inorganic and organic catalysts, clays, for instance, minerals, metallic ions, and so on. We had purines and pyrimidines, which means the potential for template-directed polymerizations, and we can synthesize without any problem, and found them also in meteorites, are membrane-forming compounds. Now, we do not know how life appeared. I have to insist on that over and over again. But here we have catalysts, which is essential in biological systems for metabolism. We have purines and pyrimidines, which is the potential for molecular replication. And we have membranes that actually are engaged in forming individual chemical systems. So you have traits of living systems formed without any problem abiotically under primitive Earth conditions. And this, the fact that we have the membrane-forming compounds is extremely important because you know that they spontaneously can undergo the formation of self-organized structures. If you have a schizophrenic molecules like a lipid, you have a head that likes water, you have a tail that hates water, and you can have monolayers, bilayers, micelles, or you can have liposomes. And the liposomes are nowadays extremely famous because you can actually add hydrating agents, and the ladies will buy them to have a beautiful skin, or you can use them in medicine to put some drugs inside cells and so on. But the basic thing is that if you have liposomes, you actually have a chemical reactor that is isolated in part from the outside. And this is precisely what the group of Jack Szostak at Harvard has been doing, forming liposomes with RNA or DNA inside and having a number of nucleotides traversing the membrane and undergoing polymerization inside. And when the liposome grows, they break apart and they can keep on growing without any problem. This doesn't mean that the liposomes are alive. It simply means that you can have, under laboratory conditions, the kind of chemical systems that could have existed on the primitive Earth, with the advantage that you are forming individual chemical systems and you are keeping all the reactants together, which is indeed a major achievement. So all the evidence that we have strongly suggests that regardless of the debates that you see among those working on the origins of life, we can form the primitive soup without any problem, with organic compounds forming on the Earth thanks to geological forms of energy, thanks to electric discharges, the energy associated with the collision of comets and meteorites. We can have different kinds of atmospheres, and now we know that we can actually synthesize amino acids even if you have a CO2-rich atmosphere instead of one rich in methane. We can have some synthesis in the chimneys, hydrothermal chimneys that you see in the seafloor. So it's very reasonable to assume that the primitive Earth had amino acids, nucleobases, sugar, lipids, and oligomers of these biochemical compounds. But the problem really, the problem in origins of life nowadays is how do we go from the primitive soup, from what we call these primitive bulabas, into something which is a central process of modern cells, in which DNA replicates thanks to the activity of proteins, and in which proteins are encoded for DNA. This is not the basis of life. This is one of the key processes of life, but clearly we have to understand how this came to be. Most people in molecular biology would not accept an explanation for the origins of life if we don't have a way of proposing how this process came to be. For many, many years, and this was again the outcome of very strong ideological debates in which on the one hand you had the highly reductionist group assuming that DNA came before anything else because DNA is a key molecule. On the other hand, some of my left-wing colleagues prefer the idea that proteins came first because, of course, they say Engels spoke about proteins, therefore they must be the first, and they tend to forget that Engels actually took that idea from a number of biologists like Haeckel, but still. So the debate was which came first, DNA or protein. And actually, we are now increasingly convinced that what came before is RNA. And RNA used to be the poor cousin in molecular biology. It used to be just a molecular go-between. Nobody will pay any attention to it. But in the 1950s and 60s, something very significant happened that I think is a lesson to everyone. In the 1960s, let me go a little bit before that. Around the 1940s, a number of very bright biochemists in the United States, in the Soviet Union, and in Belgium began looking at biochemical processes from an evolutionary viewpoint. When we speak of biochemistry today, we normally don't think in terms of evolution. But for instance, a number of people realize that if you have coenzymes, most coenzymes are actually derivatives of ribonucleotides, like vitamin B12, like acetylcoenzyme A, and so on. So many people say, what if at the beginning we had molecular processes driven by coenzymes or by ribonucleotides? And then in the 1960s, these ideas that were prospering tremendously basically came to a stop because the molecular biologists took over and there was an extraordinary tension between biochemists and molecular biologists. It's a tension that I don't know if it happened in France. I have to confess that I do not know the case of France, but it happened very strongly, for instance, in England with the people hating each other. In Mexico, it really never happened because the biochemists made the transition in a very logical way to molecular biology. But in the United States, it was a huge debate, and basically the ones that lost the debate were the biochemists, which is a pity because now we have molecular biologists that cannot not draw a biochemical pathway or do not know the structural formula for purines or pyrimidines. And they really became the ones assuming a evolutionary perspective of molecular sciences. But since the 1950s, very clearly, a number of very bright people like Brachet in Belgium or Belozersky in the Soviet Union were actually assuming that perhaps RNA had been much more important under primitive conditions. And then what is absolutely extraordinary is that in the 1960s, around the late 1960s, a number of researchers realized that if you look at protein synthesis, you see messenger RNA, transfer RNA, ribosomal RNA, and you can actually have protein biosynthesis in the absence of DNA. Not only that, but also you have that RNA can form very complicated three-dimensional structures, just like proteins. And this actually led to a number of people proposing independently absolutely independently the idea that prior to DNA and prior to proteins, there was what we now call an RNA world, in which RNA was replicating thanks to catalytic RNA that was being encoded by it. The ones that proposed the idea completely independently were Alex Risch in 1962, Carl Woese in 1967, and Francis Crick and Leslie Orgel in the same year in 2 papers published side by side in the Journal of Molecular Biology. I spoke many times with Leslie Orgel, who was a very dear friend, and with Francis Crick, and both of them told me how surprised they had been to find that RNA still has catalytic activity. They could never suspect, that indeed RNA could preserve catalytic properties after 4 billion years of evolution. But what is fascinating is to see sociologically the atmosphere that was determining the things in the time. In the 2 decades that followed the first suggestion of the RNA world, until the 1980s, many people accepted the idea that RNA genomes came prior to DNA cells. People had no problem with that. Actually, Haldane, Oparin, Bernal, John D. Bernal, and many others accepted this idea without any problem. Many accepted the possibility that coenzymes were vestiges of an RNA world, but very few people were working on it. Some people accepted the idea that histidine was a molecular vestige of the RNA world, but with one single exception in France, Marie-Christine Morel and Jacques Nignot, nobody was working on this possibility. Marie-Christine Morel is a good friend of mine, a good colleague, and I asked her a few months ago, why did you start working with histidine as a possible substitute in adenine? And I love the naive answer that she gave to me, because it was the only compound available in the fridge in the lab, so I started using it and realized that indeed histidine and adenine do have similar chemical properties. But the basic point that I want to emphasize is that no one was really working on the possible catalytic properties of RNA. No one. For a very simple reason, because everyone had been educated with the idea that in biology, the catalysts are the proteins, and the molecules that store genetic information are the nucleic acids. So everybody was being educated and trained with blinders like horses, just to see on one direction, and sociologically, a scientific reputation would have been destroyed right away if anyone had proposed an experiment or asked for money to study the possible catalytic activities of RNA. So I think it's a lesson to be kept in mind, the way that scientific prejudices limit our ability to actually explore scientifically, experimentally, evidence that is there. In any case, we now have absolutely no doubt that RNA molecules store genetic information, like DNA, and catalyze chemical reactions, like proteins. And this is an absolutely essential way of thinking in current biology that actually has some implications in the way we teach chemistry, in the way we teach biochemistry. We have to realize that the oldest component of extant biological systems is RNA, then came proteins, then came DNA, and then came processes that are involved with free oxygen and so on. Having understood that, it's very easy to see how robust the proposal is that indeed there was an RNA. We have RNA molecules that can replicate under laboratory conditions, granted, but that replicate other RNA molecules without any problems. We know that if we look at the ribosome, the catalytic site for the synthesis of the peptide bond is in the major subunit, in the large subunit of RNA, and the work of a number of people, including Ada Jonath, who was granted the Nobel Award in 2009 because of this, has shown that precisely where you have the catalytic site of the ribosome, there are no proteins. Is the RNA the one that's engaged in the catalysis? And we can have very small RNA molecules with close to 200 ribonucleotides catalyzing metabolic reactions. So clearly we have RNA molecules doing several different processes, catalyzing several different processes, and this strongly validates our idea that indeed RNA came before proteins. And if we look at the way The DNA is synthesized in absolutely all cells. If you want to have DNA, you need first to have RNA. Hence, it's very easy to imagine that once you had proteins, we had a central dogma simpler than the one named so by Crick, in which RNA was storing genetic information and proteins were catalyzing the replication of RNA. Why did we go from RNA genomes into DNA genomes? The answer is in the structure of the molecules. If you look at DNA, DNA is a molecule that is extremely stable. If you wanted to, if you were small and wanted to go inside a DNA molecule, one thing you couldn't do would be to swim, and you will be unable to swim because there is no water. The bases are hydrophobic. And if you have a hydrophobic environment, that means that the reactions of the molecules are going to be very stable. It's a very stable way that is the outcome of biochemical evolution to maintain the integrity as much as possible of the genetic information. Whereas RNA has many open loops and single strands and so on, and clearly in the absence of repair mechanisms, RNA is going to be more likely to undergo chemical changes and hence destroy the integrity of the genetic information. So we think we can understand that process as well, and I think that with that I can come to the final slide. We have an extraordinary diversity of living entities, but if we look at every single cell we know, all of them have membranes, DNA, RNA, proteins. So clearly, by the time this diversification of life took place, already cells were endowed with DNA, RNA, and proteins. From the properties of RNA and the interaction with amino acids, it's very likely that indeed protein biosynthesis emerged in an RNA world. We know very easily how to cook the primitive soup. We have no problems accumulating organic compounds in the primitive soup, but we do not know how to get from the primitive soup into the RNA world. We still do not know how life appeared. Some of us will be willing to say that this was already alive. Some of my colleagues will say, no, life is not until you have protein biosynthesis, but clearly Darwinian evolution was taking place here. Does this question mark implies that we have failed in our understanding of the origins of life? I think the answer is no, because as all of you know, in science, it's not only important to have the answer, but also it's very important to be able to pose the right questions. Thank you very much.